Dr. P. Nagraj. He is currently working as Assistant Professor in the Department of Chemistry, Anna University, Chennai. Earlier, he worked as a scientist in charge at Spices Board of India for 5 years in research and development and also in quality control laboratory. He has finished his PhD in organic chemistry at IIT Delhi. He has also done his MSc Chemical Science at Pondicherry Central University. He has published 6 papers in international journal and also participated several international events in India and abroad. He has attended several conferences and delivered lecture as keynote speaker and also undergone 12 training programs. His area of research interests are isolation, synthesis of natural products and evaluation of their biological activities, carbohydrate-based natural product synthesis, enzyme catalyst organic synthesis, green chemistry, medicinal aspects of spices and spice products.
Welcome to UGC lecture series in polymer science. Today we are going to discuss about degradation of polymer using microorganism, that is biodegradation. Today content of the lecture is introduction to biodegradation, mineralizations, microorganisms, genobiotics, biodegradations, mechanism of biodegradations and its applications. Biodegradations, what is biodegradations? Biodegradations is a chemical degradations of material by the action of microorganisms such as bacteria, fungi and algae. So, using microorganism that degradation may occur, that is called biodegradations. Biodegradable polymer is defined as degradable polymer wherein the primary degradation mechanism is through the action of metabolism by microorganism. That means, biodegradable material which will undergo degradation using microorganism, especially with metabolism by microorganism. Biodegradation is the process by which organic substances are broken down by living organism. So, that is very important. Whatever material that will undergo degradation using living organism or microorganism. The term is often used in relation to ecology. So, microorganism or living organism which is present in ecology, environmental remediations, also known as bioremediations, and waste management of plastic material due to their long lifespan. Organic materials can be degraded aerobically. Aerobically means with presence of oxygen or anaerobically, that means without oxygen or absence of oxygen. So, two way the organic material can undergo degradations. Uses of biodegradations or bioremediations. The metabolic diversity of bacteria makes them a useful resource for remediations of pollutions in the environment. Bacteria have been utilized in the cleanup of oil spills and heavy metals such as arsenic, mercury, cadmium and lead. So, using microorganism, it can accumulate all the heavy metals either in the land or water, whatever may be. Such a process is called bioremediation or biomagnification. Biodegradation is an alternative for waste disposal. That means, whatever waste which is generated, that is disposed, that is biologically that molecule will undergo degradation, that is called alternative for waste disposal. It is generally a cheaper process because we are using only microorganism, there is no other purchase of any other chemicals or any other wastage. Potentially much more efficient, using smaller amount of microorganism, it can efficiently degrade more amount of waste or polymer material or waste materials. It does not produce secondary pollutants such as incinerations and landfills. So, whatever way you go for degradations, it will produce side product. After degrade, one kind of molecule will get converted to other molecule. In case of microorganism, using microorganism, it will not give any other pollutants or landfill will not be there, safe disposal will be there. Many biodegradable polymers exist both natural and synthetic. So, biodegradable polymer may be produced in the living organism or artificially also we can synthesis in the lab, but it have to degrade using microorganism, that is called biodegradable polymers. Mineralization, the term related to biodegradation is biomineralizations, in which organic matter is converted into minerals. So, whatever degradable organic matter that will convert to minerals, that is called mineralizations. The biodegradation is a major mechanism of loss for most chemicals related to the environment. That means, after degrade it will get into the environment, but it may not cause any pollutants or any other problems if it will undergo biodegradable materials. When conversion of biodegradable materials or biomass to gases, biomass nothing but whatever waste material generated in the earth, biological living things like a carbon dioxide, methane and nitrogen compounds. So, the water, salt, minerals and residual biomass occurs. This process is called mineralization. Mineralization is a complete when all the biodegradable materials or biomass are consumed and all the carbon is converted into carbon dioxide. So, overall any organic matter after the biodegradation it will convert it to finally water and CO2, that is called complete degradation. Using microorganism, the most important organism for biodegradations are bacteria, second one is algae and the third one is fungi. These three are responsible for degradation of biological materials. These are living organisms, so which is called biodegradations. Natural polymers like protein, polysaccharides, nucleic acids all belongs to natural polymers which is present in our body. So, which is biopolymers are degraded in biological systems by oxidations and hydrolysis. So, in our body these two processes may happen, one is oxidation, another one is hydrolysis. Both way it will undergo degradations. Hydrolysis is nothing but the bigger molecule converted to smaller molecule, that is called degraded molecules. The biodegradable materials degrade into biomass, carbon dioxide and methane. In this case, if methane will generate only in anaerobic conditions, if aerobic conditions then you will get CO2 as the major product. In synthetic polymers, microbial utilization of its carbon backbone as a carbon source. Biodegradable matter in organic materials such as plant and animal matter and other substances originating from living organism or artificial materials are used by microorganisms. Biodegradable polymers decomposes into carbon dioxide, methane, water, inorganic compounds or biomass by the action of microorganism. So, in case of biodegradable polymers, the polymers normally made up of if it is a homopolymer only carbon and hydrogen. So, you will get only elimination of methane, whereas which is a heteropolymer. So, carbon, hydrogen, some other heteroatom present, and that is the case it will produce CO2 or water, something other side product. If the polymer like inorganic polymer which is made up of metals, then you will get corresponding inorganic compounds or biomass during the degradation process. The first one is bacteria. Bacteria is the important in the biodegradation process, includes there are several bacteria shown here: Pseudomonas, Klebsiella, Actinomycetes, Narcodia, Streptomyces, etc. So many bacteria which is shown here, all will undergo or it will perform degradation process either in presence of oxygen or absence of oxygen. Uses of bacteria again, Bacillus. Bacillus is a class of bacteria which is capable of producing thick walled endospores that are resistant to heat, radiation and chemical disinfections. So, this bacteria will produce thick walled endospores. So, that spores may be very resistant to heat and radiations. So, which is a very important use of bacteria. Alkali genes, again another class of bacteria which is accumulate polymer up to 90% of their dry mass. In case of wet polymer, it is a dry polymer accumulation may be done in alkali genes. Temperature is one of the most important factor affecting microorganism growth. So, microorganism which is made up of protein nothing but it is made up of enzymes. The enzymes are substrate specific, temperature specific and pH specific. So, if temperature very high, this microorganism may not be grow, only certain specific class of microorganism can survive with high temperature. Most of the microorganism will be active around room temperature, that is 25 to 30 degrees Celsius, which is activated maximum up to 40, beyond 40 degree it won't survive. Also important in the source of carbon and nitrogen and pH. Another class of microorganism which is fungi. Fungi are active in the biodegradation process. They are Sporoticum, Telomyces, these are some of the examples shown here, all belongs to fungi, and Geotrichum and Gladosporiums, Philbia, Trematus, Candida, etc. These are also belongs to fungi. Some of the fungi are aerobic, some of them are anaerobic. So, biodegradation process, so these are all the two major processes. The biodegradation process can be divided into two major classes. One is aerobic degradation, another one is anaerobic degradation. If oxygen is present, aerobic biodegradation occurs and carbon dioxide is produced. While during the degradation or decomposition and presence of oxygen, some of the bacteria or microorganism will decompose the material. It will produce CO2 as the major product. On the other hand, if there is no oxygen, that means absence of oxygen, anaerobic degradation occurs and the methane is produced instead of carbon dioxide. So, methane one of the major source. If presence of oxygen, CO2 is the major emitted product, whereas in case of absence of oxygen, methane is one of the major eliminated product, which is shown in this reaction scheme. Again, the polymer especially undergo degradations in place of depolymerases. Depolymerases is the enzyme microorganism which will do the opposite to polymerizations. Depolymerases will give oligomers, that means small polymers, and dimers, monomers, which will the polymers will undergo hydrolysis using depolymerases, which will undergo further degradations either using aerobic or anaerobic. If it is aerobic, CO2 will be the major product, whereas if it is anaerobic, methane will be major product. Another class is the genobiotics biodegradations. Genobiotics are, that means genobio means it is a man-made, which is artificial or man-made compounds. Frequently halogenated hydrocarbons, like hydrocarbons which is having halogen, those kind of compound which is artificial compound which is used as a genobiotics, that are difficult for microbes to break down in the environment. So, which is persistent of or it will withstand for long term in the environment. It is very difficult to degrade such a molecules. Biodegradation of synthetic material is complicated. So, compared to natural products, a synthetic material is slightly complicated. Suppose which is having halogen or some other heteroatom, it is very difficult to degrade compared to homopolymers, where only carbon and hydrogen present in the polymer molecules. Next is the saturated alkanes, especially saturated alkane means carbon having all 4 valency will be filled, are more susceptible to aerobic bacterial attack than unsaturated aliphatic hydrocarbons. So, aerobic attack means presence of oxygen, these saturated hydrocarbons which undergo decomposition faster than the unsaturated hydrocarbons or aliphatic hydrocarbons. Genobiotic biodegradations under anaerobic conditions such as in groundwater, sediments, landfills, sludge, digester and bioreactors, which is more favorable. In case of anaerobic, generally anaerobic bacteria able to degrade genobiotics, that means which is man-made material which is having halogens, halogenated hydrocarbons, that is easily undergo absence of oxygen, that means anaerobic condition that will undergo degradations compared to aerobic condition where presence of oxygen that may not undergo that much degradations. And they are present in various anaerobic habitats which includes inter alia sediments, water laden soils, reticuloruminals, contents and gastrointestinal contents, sludge digesters, feedlot waste, groundwater and landfill sites, where oxygen can't penetrate, and some of the landslide or martial land, those kind of places where there is no oxygen will be there. Such a place some organic material undergo degradation using anaerobic bacteria. That is what the meaning of this entire sentence. And the groups of anaerobic, some of the bacteria which is mentioned here. Anaerobes use natural organics such as proteins, carbohydrate and many other as a carbon and energy sources. Using protein and carbohydrate that will use as an energy source, it will perform the degradations. Some of them shown here: de-oleoverants and metalli reductions, acetonicums, acid overax, Botella, etc. These are all some of the microorganisms which is shown here, which belongs to anaerobic groups of microorganisms which will degrade the polymer substance. The desulfo-vibrosap, desulfomonas, microchanges, some other microorganisms shown here are major group of anaerobic microorganisms. This belongs to anaerobic microorganism. This microorganism involved in biodegradation of genobiotic compounds. Genobiotic nothing but man-made artificial compound especially which is having the hydrocarbon having halogen in their chains. So, those kind of compound normally under aerobic conditions very difficult to decompose or degrade, whereas in absence of oxygen using this anaerobic bacteria or microorganisms or fungus will undergo these kind of degradations, will produce degraded materials. Pure cultures of anaerobic bacteria must be isolated under strict anaerobic conditions, because growing and isolation is a very important field in case of microorganism. This anaerobic microorganism should be grown, that is in presence of or in absence of oxygen it you have to do. So, that condition should be very strict, then only we can able to get pure anaerobic bacteria which will undergo this kind of reaction to produce decomposition, especially xenobiotic compounds. Biodegradations of aromatic compounds. So, normally aromatic compounds is very difficult to biodegrade, but some of the microorganisms will do the biodegradation of aromatic compounds. In case of aromatic means definitely benzene or related to benzene molecule will be present in the polymer substance. So, aromatic compounds are ubiquitous in nature, which is available most of the natural product in all chemical component. Benzene ring is the most widely distributed unit of chemical structures in nature. Normally, so many natural product which is having benzene is the most commonly available aromatic compounds. Benzene, ethyl benzene, toluene, styrene and xylenes, these are all the aromatic compounds, may also be present in the natural product as well as naturally available organic materials. So, these compounds are used as fuels and industrial solvents. So, most of them used for industrial solvents for performing industrial scale of reactions. Polynuclear aromatic compounds provides the starting material for the productions of pharmaceuticals, polymers, agrochemicals. So, polynuclear aromatic compounds is nothing but more than one benzene ring fused with each other, like an naphthalene, phenanthrene, anthracene or polynucleus benzene ring which combine together. This kind of molecule have a very good application in especially in the pharmaceutical industries and the polymers industries and agrochemicals. So, aromatic compounds can be degraded under nitrate reducing, ion reducing, sulfate reducing and methanogenic conditions. So, these are all three major conditions where aromatic compound can be reduced into saturated cyclic compounds. Degradation resistant again, polymers with a pure carbon backbones are resistant to degradations, but polymers that includes heteroatom in the backbone show higher susceptibility to degradations. So, normally compared to carbon and hydrogens, suppose some heteroatom present, it is very difficult to degrade, especially polyesters and polyamides which is having heteroatom like oxygen and nitrogen, so very difficult to degrade. Aromatic polymers tend to be resistant to degradation again, despite the presence of bonds that are normally readily hydrolyzed. So, these bonds may not be hydrolyzed easily because it is a carbon-carbon bond, it is very difficult to degrade under normal condition. PET is the classic examples of such a polymer. PET polymer is particularly stable and durable polyethylene terephthalate, which is again that is generally called PET, that is which is a stable polymers. It determine that microbial communities are capable of utilizing diethylene glycol terephthalate (TTP) as a subunit of PET. So, degradation mechanism of aromatic compounds, again the aromatic compounds can be degraded using microorganism. Aerobic degradations of aromatic compounds involves in oxidation by molecular oxygen. So, molecular oxygen O2 that will react with aromatic compound, that will undergo oxidation reaction, that how it gets degraded. Oxidation leads to the production of intermediate that enters central metabolic pathways, includes the Krebs cycles and the beta oxidations. Microorganisms use oxygen during aerobic respirations to hydroxylate the benzene ring. It results in subsequent fission of the ring. Mono and dioxygenase enzymes incorporate one or two atoms of oxygen respectively into the ring. So, the major reaction is catalyzed by dioxygenases, that is enzyme that will carry out the entire reactions. Then the cleavage of the aromatic double bonds located between two hydroxylated carbon atoms are adjacent to the hydroxylated carbon atoms. That scheme reaction will be discussed in the later either it will do reaction with double bond present in the molecules, that it will do hydroxylations, either it will cleave that part or adjacent to the hydroxy group that part will be cleaved. Once the aromatic compound gets cleaved, then the degradation is easily starts. So, intermediates benzenes, aerobic biodegradation process, 3 intermediates are present, protocatachoic and gentisic acids and catechals. These 3 are intermediate during the decomposition. These compounds are broken down by similar pathway to simple acids and aldehydes that are used for cell synthesis and energy. Polycyclic aromatic compounds, toluene, xylene, naphthalene and ethyl benzenes, they are degraded by similar mechanism as benzene. So, dioxygenase initiates an attack and it produces dihydrodiol. Then the dihydrodiol is converted into catechol like a compound that can undergo ortho or meta ring fissions. Enzyme dioxygenase is indispensable to aerobic naphthalene transformations. So, the entire whatever we discussed everything will be shown in the picture. Benzene, which is a aromatic compound, react with dioxygen. This is a dioxygen. First it will form one of the double bond will undergo oxidations. So, it will get a 2 hydroxy group if both the hydroxy group present in the adjacent carbon, which is called catechol, are also known as cis benzene dihydrodiol. So, this compound is formed first, followed by what will happen, the catechol, because of this unstable molecule it will undergo aromatization, the removal of hydrogen, again you will get aromatic ring having the both OH which is called catechol. Further react with oxygen, this either it can cleave the adjacent to hydroxy group this bond or in between the hydroxy group this bond. So, it will produce a dicarboxylic acid, cis meconic acids, or on the other hand it can produce aldehyde and acids, 2 hydroxy meconic semi aldehyde. So, this kind of compound once the ring is gets broke, then further degradation will easily happen. Similar decomposition shown here in case of higher nucleic molecules like naphthalene is a higher version of benzenes. This also similarly react with oxygen to produce first 2 hydroxy groups and then followed by elimination of hydrogen will produce again naphthalene diol. This naphthalene diol will undergo further react with oxygen to produce hydroxy acids, which is again one of the aromatic ring gets broken first, followed by only benzene ring here, one hydroxy and acid group is there, namely sialic acid, which will undergo further removal of or decomposition will produce catechol. Again the catechol which we have seen in the previous slide, it will undergo degradation like similar to benzene. So, side chain group. So, whatever apart from that aromatic group, there are some side chain may be present in the molecule. Some non-oxygenated aromatics such as toluene, benzene, ethyl benzenes and xylene. Oxygenated, that means there is no oxygen present in the molecule. It can be transformed under anaerobic conditions by denitrifying bacteria in the presence of alternative electron acceptors. So, that way also we can go for decomposition absence of oxygen. In the absence of oxygen, substituted aromatic compounds are more easily degraded than non-substituted compounds. That means, in absence of oxygen, that means anaerobic conditions, substituted means some functional group like having nitrogen or oxygen, some this kind of functional group present in the molecule, that will undergo very faster decomposition than the unsubstituted or non-substituted compounds. The addition of substituent group onto the benzene ring and alternative degradations method including side chain group attack. So, the whatever functional group present on the benzene ring, that is called side chain, because of that polarity or heteroatom that will undergo susceptible to absence of oxygen, it will go undergo anaerobic decomposition. So, here shown the example substituted benzene, namely is methyl substituted benzene toluene react with oxygen, it will form first step benzyl alcohol which will undergo decomposition to produce benzaldehyde. Further it will undergo oxidation will give benzoic acid, which is react with oxygen or molecular oxygen to produce similar like catechol molecules where 2 hydroxy group present in the adjacent carbon. So, this benzoic acid react with oxygen to produce catechol like a molecule which will further undergo decomposition will produce a catechol. So, catechol again similar to benzene decomposition, which will undergo decomposition. Today summary of the lecture is, we have seen detailed decomposition, especially using microorganism, that is called biodegradations, and uses of biodegradations. Using biodegradation, what are the things we can do, that we have studied. Apart from that, biodegradation process, aerobic and anaerobic, the two main process we have seen, and aerobic anaerobic degradations, especially some of the microorganism which is active in presence of oxygen, some of them which is very active in absence of oxygen, that we have studied. Genobiotic biodegradation is a man-made artificial polymers which undergo degradations. Mostly it will be favored in case of anaerobic condition rather than the aerobic conditions, and some of the aromatic compounds, how it's undergo degradations, that we studied in details. Question of today lecture is, define biodegradations with examples, and explains about microorganism and its actions, explain the type of biodegradation process, explain about degradations of aromatic compound. Within this we conclude today lectures. Thank you.